ठीक है, so we will start with first one of you explaining to me about fed batch and continuous cultures and even fed batch, batch continuous and fed batch. कोई मुझे बताएगा? I want one of you to explain it to me. बताओ, आज तो 6-7 लोग हो खाली तुम लोग. चौधरी जयेश सतीश.
Yes, ma'am.
Tell me.
Sorry, ma'am. I just joined when you asked the question.
That is what this is the first thing that I have asked in the class. How do you explain the batch, fed batch, and continuous mode of culturing bacteria? ये तुम्हारे 133 के कोर्स में तुमने पढ़ा होगा.
Sorry, ma'am. I don't.
अरे भव्य अग्रवाल.
Yes. Yes.
अच्छा.
And there is no change from the external sources during the process, and the end
Batch
product
क्या
is
होता है?
what batch.
हाँ.
बैच मतलब कि मतलब पार्ट्स में है ना, पार्ट की जब पहले वो कल्चर तैयार होगा उसके बाद दूसरा कल्चर शुरू करेंगे, तो इसलिए बैच.
किसी और को कुछ ऐड करना है?
I guess like the batch is like a completely closed system wherein neither the nutrients are supplied nor the toxins are removed. In fed batch, like a nutrient supply is maintained, but I guess the waste products aren't removed from the culture. And in continuous process, like it's a two-way process. Like even the nutrient, a constant nutrient supply is maintained, and I guess even the toxins are continuously removed from the system.
यस विशंतन, आई थिंक तुम लोग ने इसकी पूरी मॉडलिंग भी की होगी मास एंड एनर्जी बैलेंस वाले कोर्स में, इन तीनों मोड के किए.
No, I'm not as of now.
करोगे फिर, but I'm sure the concept has been introduced in that course. Okay, and the reason why I was asking you is because whatever growth curve analysis or whatever we saw in the last class was in the exponential phase with the assumption that the system is a closed system. It's a batch system and not a continuous mode, because things will change then. All the phases, so what we talked about in the last class were the different phases, the 4 distinct phases. And what I additionally, so you all knew about the phases from your feedback. However, I tried to mention to you how each phase is significant, and how not just the exponential phase, but also the lag, the stationary, and the death phases are dynamic phases wherein you have activities taking place. So it's not just exponential, which is the most active. You will see different kinds of unique features of the cell in the other phases also. It's not that the cells are dormant in the other phases. Then I also talked about how to calculate the generation time. So the purpose of understanding the growth and the nutrients is simply because, for you as a student, is simply because you would like to mimic it in the lab. Once you understand the growth conditions and once you understand what are the requirements, it's only then that you can bring these conditions to the lab and imitate these conditions for growth of bacteria. Let me just start sharing. Okay, so microorganisms, they have been detected in a variety of extreme environments, and this figure is just a representative a cross section of Earth showing diversity of the extreme environment. So starting from permafrost or the polar regions to acid mined rains and deserts and other dry environments. If the prokaryotes, they have the potential to adapt to such a range of extremes, then for us it is important to understand the basic mechanisms by which they are able to survive these, and also, if I may put it this way, to determine the limits of life. Okay, so there are various factors which affect growth, and some of them being oxygen, temperature, pH, availability of water, then pressure, the amount of light and the type of light, everything. We'll take up the first 4 factors, the ones in blue, and see how bacteria or prokaryotes in general adapt to these conditions, how they're classified based on the requirement for these factors, and the mechanisms behind their adaptations under extreme conditions. This is just a summary table of nomenclature and the range of prokaryotes based on their requirement. Like I said, each factor will be taking up in detail. Again, repeating, please stop me wherever you have a doubt. Okay, starting with oxygen. Now, bacteria can be categorized based on their oxygen requirements or tolerance levels. So those that cannot survive without oxygen, so it's a mandatory requirement for them, they are known as obligate aerobes. And such bacteria, they use oxygen during respiration for generation of energy. Respiration here is aerobic respiration. So which is actually the most efficient mode of generation of energy when you compare it to anaerobic respiration or even fermentation. We'll be talking about all these respiration, fermentation in our metabolism classes, and where oxygen is utilized, you will see that as well. On the other extreme are bacteria which cannot live in the presence of oxygen. These are called as obligate anaerobes. So here their normal metabolism is in fact negatively impacted if there is oxygen. Then there are still others, for example, the facultative anaerobes, which can grow with or without oxygen. So irrespective of the presence of oxygen, they can grow. However, if you see in this tube here, you will see a higher density on the surface, which is only indicative of the fact that performing aerobic respiration is more efficient in terms of the energy generated. You can also use fermentation. Other normally utilize anaerobic respiration, but they are not adversely affected by the presence of oxygen. So you see this uniform spread of cells. I mean, the way they divide, they're present in uniformly in the tube. There's another category called as microaerophils, which grow. They do require oxygen, but they need it in a lower concentration. So, for example, the bacteria which you find in the gut, they are all microaerophilic. And when cultured, this is how the different group of organisms look when you see they're localized in the tube based on their oxygen requirement. This is what you see. Now, what makes them different, or what is the characteristic features of these 5 groups that I talked about? They vary in the content of 2 important enzymes which are involved in the metabolism of some toxic byproducts due to oxygen metabolism. Now, aerobic respiration, it constantly generates ROS, so reactive oxygen species, if you've heard of this. And these byproducts, they need to be detoxified for normal metabolism to continue. This is because the ROS, they are very strong oxidizing agents, and they can lead to, they can damage the cells by oxidizing other biomolecules like proteins or like lipids. And this is not just restricted to aerobes, even organisms that do not use aerobic respiration, they need some or the other mechanism to break down these ROS that are formed. There are 3 main enzymes, but this is how the distribution of 2 of them is characterized. So you will see that both obligate aerobes and facultative anaerobes have 2 enzymes, SOD or superoxide dismutase, and catalase, which is present in both. Even in microaerophile, you will see that mostly the 2 enzymes are present. However, in the case of aerotolerant, only SOD is present, while strict anaerobes, they have absolutely no mechanism of counteracting the damaging effect of ROS. So here you neither have SOD and neither catalase. Hence, the presence of oxygen is actually detrimental for this set of organisms. Now, what these do is the 2 different categories of toxic byproducts of oxygen are actually hydrogen peroxide or superoxide radicals. Now, each of this enzyme catalyzes a different set of reactions. The catalases and the peroxidases, they break down hydrogen peroxide to water. And in the case of activity of peroxidase, you will see that there's a requirement of an electron donor here, which is shown as NADH. ये तुमने biochemistry में पढ़ा होगा शायद, nicotinamide adenine dinucleotide. This is required, which oxidizes hydrogen peroxide or even other peroxides to water. Form hydrogen peroxide, and this in conjunction with catalase then generates water and oxygen. Now, like I said, obligate anaerobes, they lack all three enzymes. Aerotolerant, they do have SOD, but they do not have any catalase. In fact, there it is the peroxidase and SOD which helps counter the negative impact of the ROS. The absence of catalase is in fact used as a test for streptococci, which are aerotolerant and do not possess catalase, and differentiating them with staphylococci, which are facultative anaerobes. So facultative anaerobe will be positive for catalase and an aerotolerant will be negative for catalase. So just the presence or absence of catalase has also been used as a differentiating feature between different kinds of bacteria. Coming to the next factor, in terms of temperature requirement also, microorganisms have been classified as shown in the slide. The organisms which are isolated from, let's say, the Arctic lakes are considered extreme psychrophiles. Psychrophile, phile matlab living, loving. Psychrophiles are microorganisms that can grow at 0 degree and even below. They do not survive a temperature higher than, let's say, 10 degree or 15 degree. On the contrary, there are psychrotrophs, which are also known as psychrotolerant organisms, which prefer a higher temperature of about 25 degree and about lower temperature of 4 degree, which, as you can understand, is the refrigeration temperature. So these are of economical importance because these are the agents for spoilage of refrigerated food. Now in cold climate, because they are active at low temperature, both psychrophiles and the psychrotrophs are important in the ecosystem as decomposers. Coming to organisms that can grow at temperature of 50 to 70 degree, they are called as thermophiles. They can be found in some hot springs, and even if you look at composting piles, you will find thermophiles. And some of the classic examples would be Thermus aquaticus and Geobacillus. And I'm mentioning these two for a reason. I'll come back to it later. Higher up on the extreme temperature scale, if you see, we have hyperthermophiles, which are characterized by growth ranges of up to a maximum of 110 degree. In fact, some extreme examples can also survive above 121 degree. Above 121 degree का significance ये है कि I'm sure some of you know that an autoclave, the operating temperature for an autoclave to sterilize things is actually 121 degrees. So they can even survive beyond the sterilizing temperature that we use in labs. I've already mentioned to you that the range of growth in prokaryotes is much higher than that in eukaryotes or higher organisms. You have to understand, so basically what I'm trying to show you is the kind of range that you see here versus the other organisms. You have between, and I think somebody, one of you asked me this in one of the earlier lectures, that there's a difference between growth and tolerance. So while growth is referred to acquisition of biomass leading to reproduction, जो हमने last time देखा था कि पहले biomass accumulate होता है, cell grows to a particular size and then it divides. Tolerance means survival without reproducing. So to differentiate these two, the suffix "phile", whenever you see the suffix "phile", it is used to describe the condition under which it can grow, while the suffix "tolerant" means it can survive. So if I say thermophile versus thermotolerant, you must understand now the difference between the two. In fact, every organism has a minimum and a maximum growth temperature defining the range over which growth is possible. So this set of minimum, maximum and the optimum temperature, these three values are called as cardinal temperatures. Whenever you ask, anybody has a question? Vishantan, कुछ पूछना है?
No.
अच्छा. So when you say describe the cardinal temperatures of a particular bacteria, it means you need to mention the minimum, maximum and the optimum temperature. Now, when we discussed the plasma membrane, we talked about the membrane fluidity being affected by temperature. So at very low temperature, you will see that there is a loss in membrane fluidity, and then there's damage also due to the formation of ice crystals. There's improper functioning of enzymes. So basically here, you will see that the transport processes slow down, thereby adversely affecting the cell metabolism. While at high temperature, you see that the membranes become too fluid, and the biomolecules like proteins and nucleic acids, they start denaturing. So a cell has to adapt to these extremes, and we'll talk about what these adaptations are in both the extremes. The macromolecules in the thermophiles and the hypothermophiles are adapted to these extremes, to the high temperatures. They show some prominent differences from their counterparts in the mesophiles. And understandably so, the ratio of saturated to polyunsaturated lipids, they increase to control the fluidity of the cell membrane. Otherwise, the cell will lose its integrity. Then the DNA sequences, they show a high proportion of GC bases. The importance of GC also we discussed earlier. They are held together by 3 hydrogen bonds in contrast to A and T. And now you've covered nucleic acid, so you know about the structure of DNA. GC is in fact a marker for, let's say, strength by which the two DNA strands are associated. Also, additionally, you will find additional secondary ionic and covalent bonds, which prevents the denaturation of proteins. So basically, the macromolecules here are more sturdy to prevent denaturation. I'm talking about the thermophiles, adaptations in thermophiles. On the other hand, the proteins in psychrophiles are, in general, they are rich in hydrophobic residues. They have an increased content of certain amino acids, increased content in RNA. तुम लोग बता सकते हो प्रोलीन का क्या सिग्निफिकेंस है या ग्लाइसिन का क्या सिग्निफिकेंस है? प्रोटीन्स हो गया ना? प्रथम बंसल, आयुष वर्मा, अरे इतनी देर में तो तुम लोग ने गूगल में देख लिया होता ना. ध्वनि, हाँ आयुष बोलो.
नहीं, I don't know.
ध्वनि.
Ma'am, what I remember is that glycine doesn't have stereoisomers, and while proline is one of those that has an internal ring structure.
तो
So,
ग्लाइसिन इज द सिंपलेस्ट अमीनो एसिड, ये पता है? आर क्या
yes,
होता है ग्लाइसिन में, है ना? So whenever you will see that whenever flexibility is required, it is the glycine amino acid which provides a lot of flexibility, while proline gives rigidity. Because of this rigid cyclic structure, you will see that it is proline which contributes to rigidity in proteins. Okay, so when I'm saying that they have increase in flexibility, the proline content are actually going down in the proteins which are present in psychrophiles. And in contrast to thermophiles, here you have a lower number of secondary stabilizing bonds when compared with their counterparts in mesophiles. Additionally, you will see that there are a number of antifreeze proteins and solutes which are present in the cytoplasm to decrease its freezing temperature. And with respect to membrane lipids, तुम लोग ही मुझे बता सकते हो, psychrophiles will have more unsaturation, which will result in an increase in fluidity because you do not want a gelling of the plasma membrane. The growth rate of these prokaryotes, they are much slower than that under mesophilic conditions, and this is also a kind of adaptation for organisms in the extremophilic conditions. Okay, coming to the next factor, which is pH. Now, similar to cardinal temperatures, the microorganisms also have cardinal points in terms of their pH range. Mostly bacteria are neutrophiles, meaning they can grow optimally at a pH within 1 or 2 pH units of the neutral pH. But there are pathogenic strains which are resistant even to the acids in the stomach. क्या condition होती है? Do you know what is the pH in our stomach? Anybody?
Mom, it's around 5 to 6, right?
नहीं, gastric
Then
acid
3 to 5.
correct, even lower, 2 भी होता है. So it depends on which part of the gut you're looking at, but the pH of gastric juice ranges from 1.5 to 3.5, and you have organisms surviving under those conditions. Okay. So in comparison, if you remember, ये तो bacteria के लिए I was saying that they prefer mostly they are neutrophiles. In comparison to these, if you remember what I told you about fungi is that they prefer slightly acidic values. So 5 to 6 is the optimum for fungal species. Microorganisms that grow optimally at pH less than 5 are called as acidophiles. And extreme acidophiles you can even find, other than the ones thriving on the gases, you can even another component of the normal microbiota, which is lactobacillus, is also tolerant to acidic environment. Now, when I say phile and tolerant, you have to understand कि growth नहीं कह रहे हैं, tolerate करने को कह रहे हैं. And the lactobacillus have a range of tolerance of about, it's a large range, 3.5 to something like 6.5. In fact, lactobacillus also contributes to the acidity by producing lactic acid. Talking of alkali phials, the best example is of the pathogen Vibrio cholerae, which is the pathogenic agent of cholera, which can grow best at slightly basic pH of 8, but it can survive pH values of even approximately 11. Just a list of items which we commonly come across with their pH and how increasing acidity or increasing alkalinity. This is just a scale to show what acidophiles and alkali phials can thrive on. How do they survive? These extremophiles survive. So starting with acidophile, the proteins in this category of prokaryotes, they exhibit higher negative surface charge for stabilization at low pH. Then they have pumps operational for expelling protons out of the cell. There are special chaperones to protect their biomolecules like DNA and proteins. Now, have you heard of the word chaperones, students? If not in science, maybe in normal English literature. Chaperone सुना है किसी ने? नहीं सुना है? Chaperones, हाँ बोलो ध्वनि.
Yes, I'm basically somebody to overlook something, maybe.
Correct. So ये in during old times these were adults, especially to overlook when two young people are meeting. Okay, they were kind of mediators or the ones supervising these young people, especially when there's an alliance that's being sought. Okay, so these special chaperones in the molecular terms are proteins which aid in correcting or rectifying misfolding of proteins, or just to protect these biomolecules like DNA and proteins. And chaperones are a kind of a general mechanism adopted by some of the extremophiles to protect these biomolecules. Then we talked about the tetraether lipids in archaeal membrane, which are also known to contribute to the resistance towards low pH by being highly resistant to proton permeation. So this is what you want to control. Whatever proton is inside has to be expelled out to bring back the cytoplasm to normalcy. And the cells also contain some buffering molecules which have basic amino acids to sequester the protons. In contrast to acidophiles, alkali files, on the other hand, they have mechanism of pH homeostasis in the cytoplasm by using a number of pumps. And in some bacteria, there are certain secondary the cell envelope. It confers a means of adaptation to high pH. Then the cell uses deaminases to produce acids that lower the pH of the cytoplasm. So it's just acting counter to, so if it's alkaline conditions outside, it has to bring down the conditions. So maybe this is a means of adapting by producing acids. Their proteins, the alkali files, the protein profile that they have, they are adapted to functioning at a high pH. That is, they are pH stable compared to their counterparts in the neutrophiles. And such sodium antipodes also help in bringing in protons to bring down the pH of the cell. Having told you about the composition of the prokaryotic cell in one of the earlier lectures, I need not re-emphasize on the importance of water for microbes. The availability of water for a cell depends upon its presence in the atmosphere, that is relative humidity, or its presence in a solution in which the cell is growing. This is called as water activity. Now, water activity of pure water is 1, that's 100% water. It is affected by the presence of solutes dissolved in water. So you can imagine that higher the solute concentration of a substance, lower is the water activity, and vice versa. Prokaryotes can actually tolerate a range of AW from about 1 to 0.7 normally. The only common solute in nature that occurs over a wide range is actually salt, NaCl. And in fact, if you look at the saline environments, they make up a large proportion of the earth, from the marine environment where you have 3 to 4% salinity to even hot springs where you have as high as 10.5% salinity. And this is just an example to show you how the water activity changes with the increase in solute concentration. The microorganisms are categorized based on their growth response to salt. Those that require NaCl for growth are called as halophile compared to the non-halophile, which do not require salt. Mild halophiles, they require 1 to 6% salt, while moderate halophiles require 6 to 15% salt. However, there are also extreme halophiles which require 15 to 30% NaCl for growth, and these are mostly archaea. Bacteria that are able to grow at moderate salt concentration, even though they grow best without salt, are called as halotolerant. So you see this green curve here. Now, different authors have categorized it. They've made further classification and further subgroupings, extreme, halotolerant and stuff. You go into the details. Non-halophile, that is the subfigure A, if you see, in case of high salt concentration in the environment, due to the flow of water out of the cell loses its integrity. So the adaptive mechanisms that halophiles have, they either adopt one of the two strategies, either a salt-in strategy or a osmolyte strategy. Now, moderate halophiles, they maintain their structure by synthesizing compatible organic solutes called as osmolyte. Now, this is what is referred to as osmolyte strategy. These osmolytes protect the cell proteins from denaturation in water that contains salt. While extreme halophiles, they maintain their structure by employing pumps to maintain an equilibrium in salt concentration intracellularly, and that in the environment. So this is there's an extensive adaptation of the intracellular macromolecules to function under such high salt concentration. So you see these chloride ions being taken in. This is known as the salt in strategy. They're bringing in salt to make up for this, to maintain this equilibrium. Now, several structural changes take place, like having charged amino acids on the surface, which allows the retention of water molecules around these components. That was about the 4 factors. So this is just a summary slide of various adaptations under different kinds of extremities, wherein you will notice that there's some common adaptations, like you will have DNA repair proteins, which are found in most of these extremophiles. These are required for DNA integrity, maintaining of DNA integrity. Then there are also chaperones that you will come across. Like I said, they happen to be a general mechanism of adaptation which aid in correct folding of protein for their functionality. So they act as basically protectant for against denaturation or damage of proteins. In many cases, you will also find the pumps to be very helpful in maintaining homeostasis. Now, the reason why we've been talking about adaptation in extremophiles is, of course, one thing that you try to understand the limits of life and you try to understand their mechanism to be able to mimic it. But why do we want to mimic it? Because there are used. So extremophiles serve as a source of various biomolecules. Besides, they're also commercially exploited in some of the biotechnological processes, like in treatment of waste or even in bioremediation, or let's say agriculture under extreme conditions. Hence the need to actually study them and study the different factors and the mechanisms which are involved in adaptation. So after, if there are any doubts, please let me know now, because we'll now go to studying the growth. How do we study them? कोई इसमें doubt किसी mechanism में? I just talked about 4 factors. You can go through the other factors if you want. Prescott has all these details very nicely mentioned. So अभी हमने temperature, oxygen, pH और water availability देखा, और कैसे extremophiles adapt करते हैं इन चारों extremes में. ठीक है? If there are no doubts, then we go on to the measurement of growth. So how do you know if cells are growing or cells are dividing? Either you can see that or calculate that by direct counting, which can be done. So here you measure the cell number or the cell weight, or there could be various indirect means of measurement of cellular components, like if you measure the total N or the total protein. अच्छा, तुम लोग मुझे ये बताओ, तुम लोग के बायोकेमिस्ट्री के प्रैक्टिकल्स की थ्योरी भी थोड़ी बताई जा रही है क्या तुम लोग को बायोकेमिस्ट्री में? क्योंकि ये सब तुम लोग के एक्सपेरिमेंट्स हैं बायोकेमिस्ट्री में.
नो मैम, अभी तो मैम बेसिकली मेन बायोमोलिक्यूल से चल रहे हैं, लाइक प्रोटीन्स.
हाँ, तो बायोमोलिक्यूल में जैसे तुमने प्रोटीन किया, तो इसका एस्टिमेशन वगैरह पढ़ा है क्या?
नहीं मैम, वो अभी बाद में है, अभी तो खाली स्ट्रक्चर्स किया था.
अच्छा अच्छा, ठीक है, बाद में. लेकिन हम ना बताते जाएंगे कुछ प्रैक्टिकल्स की चीजें. तो जैसे इस समय भी हम तुम्हें बताएंगे ये सारी चीजें आप प्रैक्टिकल में माइक्रोबायोलॉजी में भी करोगे. सो आई विल गिव यू वेरी बेसिक अबाउट व्हाट इज हैपनिंग. अदरवाइज द experiments happen parallelly, so it's quite easy for you to understand कि theory के साथ साथ practical. लेकिन मुझे पता है कि जब next कुछ महीनों बाद होगा तो तुम लोग सब भूल चुके होगे. ठीक है. So indirect measurement, I was saying, like if you quantify the total N, or if you quantify the total protein, DNA, ATP, the certain metabolic processes, like if you assess O2 production or its consumption, yeah, nitrogen production or consumption, it will talk about activity, about growth, about how cells are dividing, or whether or not the cells are dividing. If you see increase in protein, you understand the cells are dividing, right? So these are indirect measures of cellular components. Direct counting can be done using a hemocytometer or a counting chamber. The hemocytometer is a simple gridded system divided into various cubes for accurate counting of number of cells, and then you can back calculate the density or the concentration of cells in a sample. So what you see here is called as a Petrov-Hauser chamber. So this is this slide that I was mentioning for counting the bacterial cells in a measured volume. In fact, if you see in this enlarged picture, you will see that these red things are actually RBCs in a fixed dimension. So you, for example, you can calculate 4 different cubes and the number of cells, and then you go on to calculate, to back calculate the concentration in the original sample. The grids basically, they facilitate in precise counting. Uh, but you might imagine that if you do a counting of this sort, you are not able to discriminate between live cells, dead cells, and even sometimes you can get confused with the debris of the same size when you view it under the microscope. So you basically load your sample onto this gridded slide and view it under the microscope and make these countings or count these cells. But like I said, in such a simplistic slide calculation, you will not be able to differentiate between dead and live. So now there have been refinements in the sense that certain fluorescent stains are used in these slides to make it possible to distinguish even between live and dead bacteria. Um, Another technique uses an electronic cell counting device. So the earlier one was completely manual. Here, there's an electronic cell counting device, like for example, the Coulter counter, which is based on detection and counting the changes in electrical resistance in saline solution. So an electrolyte containing the cells travels through a small aperture, and the 2 electrodes are placed such that resistance can be measured. So as the cells pass through the small aperture, there's a brief change in resistance which is measured between the 2 electrodes, and this change is recorded by a sensor. So each resistance chain represents a cell. Theoretically, it represents a cell. Now this method is rapid and accurate, but if the culture is too concentrated or if the cells pass through the dilution that you use for measurement of the number of cells in a particular sample. Again, this method also does not differentiate between live and dead cell. It's only seeing the change in resistance as the cells pass through this aperture. Another method by which cells can be counted, and here you can restrict to only counting the live cells, is by allowing the samples to grow on a solid medium. So if this is the suspension of bacterial cells and you would like to count the number of cells in this, you can simply take a volume, a known volume, and you can spread it onto the solid medium. If sufficient mixing has been done and the cells are not in clumps when you spread them on the medium plate, each cell will give rise to a single colony. So for visualization, if the single cells are plated, you will not be able to visualize it. But once you allow an incubation time, each cell will give rise to a colony of this sort, which can be counted. Again, in this case, you have to be sure that the dilution is appropriate so that each colony originates from a single cell. While here, what you get is an estimate of the live cells, and the method is also quite easy. It is the slowest from whatever method that I talked about, the Coulter chamber or the hemocytometer, because you're giving a time for incubation, for growth, or for the colonies to be visualized. Yeah, that was about counting the cell number. Now there's several methods for measuring the cell mass as well, including using ordinary balances to weigh a sample. So let's say you want to weigh the dry weight and you want to track while monitoring a bacterial growth curve, if or not the cell is growing, you can simply sample at different intervals and see if the dry weight is increasing after the water has been removed. Another commonly followed method is by using a spectrophotometer. This is also something which you will be using quite often for your practicals in biochemistry, wherein you estimate, let's say, DNA or proteins. This requires very little sample volume, and spectrophotometers are basically equipments that can measure turbidity accurately. So the culture is placed in a translucent cuvette, and the cuvette is placed in the machine. The turbidity is measured immediately. By basic calculation, these can also be converted to cell concentration, the turbidity value. अगर Beer-Lambert's law पढ़ा है तुम लोगों ने तो. This method of turbidity measurement is better known as the determination of optical density, OD measurement. And the more cells are in the suspension, the more light is scattered or absorbed. and less light can be detected. So this is a method which is cheap and its operation is fast and easy. Hence, this is a choice for a quick measurement of bacterial growth. You will see that most of the labs and most of your experiments will also involve the spectrophotometric measurements. Like I said, other than all this, you can measure the uptake of substrates or the formation of product. Various specific assays can be used for detection of a particular moiety. One way is to measure the biomass. One way to measure the biomass is actually by measuring ATP. So ATP, you know, is only present in live cells. So if you're able to quantify the ATP, you can see how many cells are growing or what is the rate of growth. Now, I've tried to outline some of the very basic techniques, though now we have some very sophisticated method, like including even online probes. So you really don't need to sample and then subject it to such kind of methods. Various molecular biology tools are also helpful in this. Maybe in one of the advanced courses, you will be informed of some of these other techniques, the higher end techniques. What I mentioned to you was only related to what you will be doing in the practicals in general microbiology once we have the practical component. Yeah, so this I'm now open to questions, students. Let me know if you have a doubt. किसी को कुछ नहीं पूछना है? हाँ बोलो ऋतुराज.
While measuring the optical density, it gives the value for the both dead and living living
Yes.
cells.
Yes,
Okay.
yes, rightly pointed out. This is also a method because you're only measuring turbidity.
Yes,
And
ma'am.
अगर तुमने growth curve वाला lecture सुना होगा, I don't know if you
Yes, ma'am.
read that. I mentioned that even during the decline phase, it is not that the turbidity is going down. ठीक है?
Yes.
Because ये live और dead के बीच में differentiate नहीं कर रहा है. It's only measuring the increase in turbidity, and dead cells में भी turbidity रहेगी, है ना?
Yes, ma'am.
So this is out of all the methods that I talked about right now, the one which was cell plating, which is the only method which gives you a live count. Otherwise, you can use a stain in one of those visualizations, है ना? Fluorescent stains अगर कुछ use करो तो कर सकते हो.
Thank you.
Anybody else has a doubt? No, everything is clear. Good, then I would like to stop here because in the next class I'll try to start talk about different methods of isolating bacteria. Okay, we talked about growth right now, and then we'll talk about isolation and cultivation and also preservation of bacteria. ठीक है, then we'll stop the class. See you all students in the next class.
Thank you.